house.
Breakdown of hydrogen peroxide by Catalase. The main focus of our lab was to find the rate of reaction for potato catalase.
Background: enzymes are biological catalysts. They help to increase the rate of chemical reactions. Enzymes have a 3D shape which makes them highly specific for the reactions that they catalase. Catalase is the breakdown of hydrogen peroxide.
Introduction. Our lab revolved around mixing hydrogen peroxide with catalase to find the rate of reaction. First, we used controls to find the optimal pH. Our lab was conducted by varying pHs to find the best environment. After 60 seconds, we measured the height of the bubbles. Then we found the volume of bubbles to determine the rate of reaction.
First, the water is being measured. The potato was ground in the mortar by the pestle. The potato was transferred into the test tube with the water in it. The pH was controlled by adding acid. The reaction is started and mixed. And the height of bubble is measured.
The graph right here represents the pH 3. On the right side, as you can see, represents the height of bubbles, and on the left side represents the trial number. The average height of bubbles was 0.1006, while standard deviation was 0.052. The standard deviation is comparatively large relative to the graph, which is why there are a lot of curves on the graph. On the perfect graph, the line.
The table on the right shows the trials and the height of bubbles per second. Trial 8 was omitted from the average score because it was too low, being 0.05 compared to the other trials being around 0.2 to 0.3. The graph on the left shows the reaction rate, aka height of bubbles per second, on the y-axis and number of trials on the right x-axis.
This shows the pH 10. On the right shows the trials with the height of bubbles per second, and the graph is supposed to be a straight line, and as you can see, it's not a straight line. This is due to varying height of bubbles per second, which either go back to inaccuracy in the measuring of potato or inaccuracy of the amount of hydrogen peroxide put.
Next, we compare the different pH environments to find the optimal pH. As you can see, pH 3 had the lowest number of bubbles per second, while it steadily increased all the way until pH 10. This simply shows that pH 10 had the most acidic environment, which was the most acidic environment, also had the highest average rate of bubbles per second. The optimal environment of pH 10 far exceeded even the environment of pH 3 by a total of 3 times the amount of bubbles per second. So to find the calculations for volume of bubbles for the pH 10, we were going to use the equation pi r squared times height. So we're giving the average height of bubbles per second for pH 10, which is 0.30674, and we multiplied it by 60 to find the average height of bubbles. The average height of bubbles for pH 10 was 18.4044. The equation used for V is pi r squared times height. So plugging in these variables, we have 3.14 times 9 pi r squared times 18.4044 millimeters, which yields 4,680.925. We take this and we divide by 60 to find it per second, and we find out that the average volume per second is 78.0163.
The published result for potato catalase is 7.0. Some improvements that could have been made to improve this experiment was to measure the bubbles in increments of 16 seconds rather than after 60 seconds. Had we have done this, we would have been able to find the spike of the reaction and when the hydrogen peroxide was no longer available. Why was our optimal pH wrong? It was wrong because of the high rate of standard deviation within our results.